So I was talking about H4K12. Why would one acetylation site would be so important? So there was a paper coming out last year which was trying to figure where histone acetylation sits on a coding region of genes. So all the histone acetylation have the same trend. If you look at the promoter area, they tend to cluster and peak within the proximity of the promoter, and then it goes down. Except from one acetylation site. Again, this guy, H4K12. So this guy peaks a bit in the promoter area, but then it stays constantly represented through all the coding region until the very end. So this is very, very specific acetylation site. And it's also actually been shown in aging that in oocytes it's also deregulated. And it's the only one that deregulated. So we tried to figure out what's now the connection between our gene expression and the H4K12. And to this end, we took these two groups of mice, the young and the old field condition. We took the hippocampus out, and now we try to sequence and see where this histone sits and what will be the difference between these ages. I mean, the question is, will it be like that or something strange will happen? So two likely mechanisms emerge from this experiment. First one that can affect gene expression is the elongation process of the RNA polymerase. And if you look here, then you can see that in the young ones, if you start the beginning of the promoter until the end of the gene, you see more or less representation of this H4K12 throughout all the gene. But in the old guys, there are two things. There are major gaps, and also there seem to be many, many more peaks, so it's like not evenly distributed anymore. So there's some sort of deregulation after 1 hour field condition. And you can see it here, it's significant, so you see that almost 65% is covered with this H4K12 in our gene array genes that we found in 2000, and there is much less in the old. When you look at non-coding regions, this effect is gone, so there's almost nothing of this guy. The second is initiation, so again, it can be also a difference in the promoter area. And sure enough, we found that 100 of our genes in the gene array, you have more clustering of H4K12 in the promoter area, and this difference in this 100 genes is much more, they are much more in the young ones than the old guys. And now we wanted also to go a bit deeper, and we took one example, and we took the forming gene, which is just, to keep it very simple, is a regulator and nucleator of actin, which was very dominant pathway in our gene array. So we look at this guy, and now we'll take a magical journey with this guy. And we'll start with the DNA, and in the young ones there's more H4K12 after 1 hour field condition, and we don't see it in the 16 months old. This reflects on the levels of the mRNA, so there is much more of expression of this protein. And in turn, it obviously will affect the protein levels, so you have much more of this protein after 1 hour. And interestingly enough, by coincidence, we have a knockout of mouse of formin 2. And you can see they somehow have a phenotype of accelerated aging. So already after 8 months, they freeze much less than the 3 months old if they lack this protein. So we can nail it down to one protein. Another question is, can we affect the whole machinery on all the other genes? So if you remember this slide, I showed you that if histone is acetylated, it opens the chromatin structure. And we have drugs that can reverse, which can block this reverse process. So we can artificially increase the acetylation. And to this end, we took mice, we injected the cannula in the hippocampus, and we injected SAHA, which inhibits those histone deacetylase, which are called HDAC. And sure enough, we found that after 1 hour field condition, the SAHA guys have a trend that you can see that there is more histone acetylation, but again, strangely enough, the only significant site is H4K12. And this leads to, not only now in the formin 2, but also in many representative genes, an increase in H4K12 in various regions. And sure enough, it also causes elevation of gene expression. But most importantly, mice that receive SAHA 1 hour before training freeze more and show better associated memory. So, just to conclude this very fast talk, I'm sorry if it was too fast for 10 minutes, not that enough. So in early aging, we're not talking about really late aging, when you just have beginning of declining your memory function, there's some sort of deregulation of specific site, H4K12, which affects the gene expression profile, and therefore it leads to some cognitive deficits. And well, we showed partially that if you use a HDAC inhibitor, you can elevate H4K12, and you can sort of get a normalization of the gene expression, and therefore you have improvement in cognitive function. I just want to thank everyone that helped me in the lab, and especially my boss, Dr. Arnold Fisher, who is not only a brilliant scientist, but also can cope with my crazy personality. And I would like to thank you for your attention. Thank you very much.